Good morning. Well, good morning from the UK. I guess that wherever you are, wherever you've joined the event from, the time will be rather different. So, welcome to this International GCSE Human Biology training. As Jaira said, my name is Paul Jackson. Um, I have taught in the UK for 40 years. I have been a senior examiner for many, many years. I have um, been involved with writing UK GCSE biology papers, all of the associated work with marking papers. So I have a very, very wide range of experience, which hopefully, as we go through this presentation, I'll be able to give you some insight as to how specifications are developed, information about the exams, the questions, the mark schemes, etc. As Jaira said, if you have any questions, please put them into chat. But should you wish to sort of ask a question directly, you do have the opportunity to unmute your microphone should you wish to ask a question. As we go through, if you do have any questions, please put them into chat in the first instance. I do have a separate screen, so I'm monitoring the chat so I can see immediately if you choose to put something in there. And I'll try to answer questions as we go along rather than sort of save them all up until the end. Okay, so as I said, my name is Paul Jackson, and I've worked for Pearson for many, many years. I have taught the UK human biology in the past, and the international GCSE human biology is very similar to in terms of the assessment, etc., to the international GCSE biology. Okay, so in terms of this professional development, it's designed for teachers who are looking to teach this human biology specification. We'll be looking today at planning and teaching the course, understanding the assessment and preparing students, giving you some understanding of how the content is assessed by looking in particular at assessment objectives and mark schemes, looking at how Pearson mark schemes should be used. Different awarding bodies use mark schemes in slightly different ways. I mean, in general, mark schemes across awarding bodies have lots of similarities, but there are specific things associated with each awarding body. And we'll have a look towards the end of the support that's available to you. So in terms of this training, it is aimed at people who are sort of looking to get started teaching this specification. Perhaps if I give you a couple of minutes to have a think about these questions, and perhaps just put a few comments in chat so I get a feel for today's audience. So, have you been teaching it already? Are you looking to teach it at some point in the future? Have you just started teaching it? Are you familiar with Pearson qualifications? I mean, for example, do you teach the international GCSE biology or the international A level? And perhaps what are you hoping to take away from this session today? So the chat is open. I'm monitoring it. Perhaps to say, if you could put something in there, just give me an idea of today's audience and the breadth of experience. Tell me how long you've been teaching, that kind of thing. Hello. Okay, so we have one comment. So someone not teaching the course yet, but a former IG student. So obviously some personal experience of Pearson qualification. So thank you for that. I'll just wait another 30 seconds or so, see if we have any more comments before we continue. As I said, if you do have any questions as we go along, please do put them into chat. And as I said, I'll try to answer any questions as we go through the course. Okay, so let's move on then. So as I said, welcome to Pearson, or Pearson Edexcel to give the company the full title. Pearson is the world's leading company, or leading learning company, I should say, and the UK's largest awarding organisation. So in the UK, we have several awarding bodies. Pearson is the UK's largest, and they're best placed to provide qualifications which are aligned to the British educational system. Pearson has a heritage stretching back over 150 years. So obviously things were rather different 150 years ago, but the international heritage is very long. And Pearson partners with schools, universities, and employers worldwide, offering globally recognized qualifications to a huge number of students every year, so approximately 3.5 million. And there are a large number of trusted and recognized qualifications partners to about 6,500 schools, colleges, and employers globally. So Pearson reaches out far and wide. In the UK alone, each year, approximately 10 million exam scripts are marked on behalf of the UK Department for Education. So obviously that's a huge number of scripts over many, many different types of specification. And Pearson operates in a very large number of countries, so approximately 70 countries worldwide. So just coming back to the comment in chat, someone just commented that they're looking forward to teaching this course because they're a biology major, and someone just praising Pearson for the support that Pearson offer and the help given in teaching and preparing students for GCSE. So thank you for that comment. Okay, so in terms of the international GCSE human biology specification, we'll start off with an overview. We'll have a look at some of the content, and we will focus on assessment. We'll focus on assessment objectives. So there are 2 exam papers, and those exam papers both contain a range of questions. So there's short structured questions, there are multiple choice questions, there are questions involving data handling, graphs. There are extended response questions. So the extended response questions are questions which usually have a mark ranging from 4 to 6. So as we go through, we'll have a look in more detail about these question types. There are clear mark allocations and topic coverage outlined for each paper. There are 12 topics in the specification. So, and there are details for each topic, which should help you to sort of break things down as you do your teaching. Assessment objectives, there are 3. AO1, which is recall of knowledge and understanding. AO2 is application of knowledge and understanding, and AO3 is analysis and evaluation. And AO3 encompasses questions which involve practical work. And there are there is a sort of a framework in terms of percentages for each assessment objective and the assessment of them. I have a question about recording of the training. I can't give you a direct answer to that, but I think our Pearson host might well respond to that in chat. You should have either received already, and our Pearson host has put the link in the chat. You can obtain the slides for today, but in terms of the recording, I am not 100% sure. So hopefully our Pearson host will put a response to your question in chat. The specification also contains a taxonomy of the command words. So there's a glossary telling you what each command word involves. And again, we'll have a look at that in more detail as we go through. So typical command words in questions are describe, explain, evaluate. We use command words such as state. We use command words such as draw, calculate. So as we will look at this in more detail, but there are a large number of command words, but some of them are used much more frequently. In terms of mathematical skills, students will be tested on graph work, ratios, percentages, possibly some geometry. And math skills make up approximately 10% of the total assessment. Practical skills are assessed through the written exam papers, not through any practical assessment. There are core practicals in the specification, and these are they usually stand out because they are italicized. And it's important that students understand methods, how to record results, how to deal with results. They need to be familiar with controlling variables, identifying variables, and they need to be mindful of safety considerations. Just coming back to the question about recording, our Pearson host has put a response to that, so thank you. The recording will be available after the event 24 to 48 hours in the same link that you entered the lobby. So that's that question answered, so thank you for that. And the other important thing about this specification, as with other Pearson specifications, is that the idea is that students develop transferable skills which involve scientific thinking, data literacy, sort of reasoning, communication skills. So there is a section in the specification about transferable skills, which is an important aspect of these specifications. When you go online, the Pearson website, and identify the human biology page, you can download the specification, which I'm sure most of you have done. The specification currently is on, as far as I'm aware, it's on its second issue, which I think was done or published last year. Near the front of the specification, you'll be able to find a small table which tells you any changes from the previous version of the specification. But currently, as far as I'm aware, the specification is issue 2. So the specification was published in 2017, with the first assessment in 2019, which is the same as other Pearson GCSE sciences. From the Pearson website, you can access all sorts of information. Again, we'll come back to this later, but course material so you can access the specification, sample assessments, lots and lots of exam materials are available to you. So past papers, the mark schemes, and examiner reports, which I'll talk more about later. So there's lots of material available. You can also access past training and professional development, which Pearson have provided. So there's lots of support, and so we'll focus on that towards the end of today's presentation. Okay, so we'll have a look at the content in a little bit more detail. So as I said, altogether there are 12 topics. Some are larger than others in terms of the content, as you will perhaps have noted if you've actually looked at this already. But the content varies for each topic. So topic one is obviously like it's a bit of a building block topic in a way. So we're looking at cell structure and function. We look at genetics. We look at sort of organization from cells to tissues to organs. So students need to be able to identify organelles. So perhaps look at an image from a light microscope or a transmission electron microscope. They need to be able to identify organelles such as mitochondria and ribosomes. And they need to be able to give the functions of these organelles. They need to know about DNA and protein synthesis. So the fact that DNA is a double helix with complementary bases that are paired, that genes code for proteins. So the fact that 3 bases are used to code for 1 amino acid. And then they need to know about protein synthesis and the role of RNA in transcription and translation. And then slightly more challenging aspect of the topic, mutations, genetic engineering, sort of changes in bases, phenotype changes, looking at GM organisms used in, for example, producing insulin and vaccines. So obviously in terms of things like COVID, obviously that's not in the specification, but that's sort of a good example of the use of genetic engineering in science, in biology. And also looking here at sort of genetic engineering in nutrition in terms of genetically modified organisms and food production. So continuing with topic one, students need to know about mitosis, stem cells, so sort of basics of the stages of mitosis and the fact that 2 genetically identical diploid cells are produced. Stem cells is something that students often find a little bit more difficult understanding about the role of stem cells, but they need to know about adult stem cells, embryonic stem cells, the fact that these cells can specialize, and they need to consider ethical issues related to the use of stem cells. Tissues and organs, so students need to learn about the formation of tissues from cells, examples such as muscle and bone. They need to learn about specialized cells such as egg cells and sperm cells. In terms of assessment, it's useful to use diagrams, obviously label diagrams of cells, annotating perhaps to give the functions of the different organelles. Students need to be able to learn the function of cells, so sort of checking that they can give a definition, checking that they can give perhaps a brief explanation of the function of a cell. So topic one is quite heavy on terminology. There are some sequencing involved, such as the stages of mitosis. There are processes to describe. Protein synthesis being one, which is perhaps a little bit more difficult. Genetic engineering, another process, which involves some sequencing. So the use of diagrams, perhaps animations, even sort of animated videos, etc. Using resources like that will help students to sort of learn some of the content of this topic. So moving on to biology, topic 2, which is biological molecules. So students need to learn about the structure and function of biological molecules. They need to learn about enzymes, how enzymes support life processes. They need to learn about specific molecules, so the structure of carbohydrates, proteins, and lipids. They need to know about the subunits that carbohydrates, proteins, and lipids are built up from. So again, the use here of sort of diagrams is important to help and support learning. Some practical work, so food tests. Students need to learn the tests for glucose, starch, lipids, and proteins. So obviously hands-on practical work is important here. And there are some core practicals related to this part of the specification. So core practicals which investigate the vitamin C content of food, and core practicals which involve the use of enzymes. So there is a good opportunity for practical work here. In the previous topic, in topic 1, I mean, some practical work might relate to perhaps some microscopy, just looking at cells using a microscope. In terms of enzymes, students need to learn that they are biological catalysts, and they need to understand how they're affected by temperature and pH, substrate concentration, and inhibitors. So again, for some students, being able to explain how these different factors affect enzyme action can be challenging. So there are some areas of difficulty in this topic. The use of immobilized enzymes, so lactose-free milk production, converting sucrose into other sugars, and diabetic test strips. Again, conceptually, some students find this aspect of the topic quite difficult. One practical that they could use would be the preparation of alginate beads, which contain an immobilized enzyme, which is then used, for example, to remove the lactose from milk. I appreciate and understand that the facilities that you may have for doing some of this practical work may be challenging. So that's why perhaps using videos of some of these practicals with your students would be useful. And in terms of assessment, there's lots of application of ideas involved here. So the opportunity for students to be able to perhaps consider contexts which they're unfamiliar with, and to apply some of their knowledge and understanding to those unfamiliar concepts and situations. So topic 2, regarding biological molecules, again, there's some very important biology here. And there's a range of difficulties in terms of the content of the topic. Topic 3 is about the movement of substances. So again, some important biology which can be applied in many, many different situations, both in plant biology and obviously human biology, animal biology. So students need to know about diffusion, they need to know about osmosis, they need to know about active transport, and they also need a basic understanding of water potential so that they can explain osmosis accurately. So this particular topic and the processes that we need to learn about here, the content here lends itself to questions which are based on perhaps unfamiliar situations, and students have to apply their knowledge. Obviously, some recall as well, sort of learning definitions of the processes, but also there are some really good opportunities for sort of checking students' understanding by asking them to explain a situation that's unfamiliar to them, but they just have to apply what they've learnt about the content. Students need to learn about what kind of factors affect the movement of substances into and out of cells. So they need to be familiar with surface area to volume ratio. They need to know about how temperature affects these processes. They need to know about concentration gradients. So, as I've already said, one way in which assessment is focused here is on application to unfamiliar situations, such as placing plant cells or animal cells in different solutions. So perhaps a sugar solution, a salt solution of different concentrations. And the students need to understand what could happen to these cells in these solutions and explain what happens in terms of talking about diffusion or perhaps osmosis in particular. Topic 4. So some biology here which we would not find in a IGCSE specification, but specific to the human biology specification. So they need to know about the structure and function of the skeleton. They need to learn about joints and muscles and the role of these structures in the movement of the body. So they need to know specific details of the skeleton, the axial skeleton and the appendicular skeleton. They need to know the names of these bones that we can see on the screen. And they need to know the structure of a long bone. So clearly sort of label diagrams here, good for learning, good for testing. Joints, so students need to learn about the types of joints. They need to know some examples. So we've got some specified examples, the elbow, shoulder, intervertebral discs. And they need to know the specific features of a synovial joint. So again, sort of using diagrams that can be used for part of the learning and also the assessment, sort of labeling a blank diagram of a joint, of the skeleton, really good opportunities for testing knowledge and understanding. So in terms of movement and muscles, students need to know about voluntary muscles, specifically the biceps and the triceps, and how these muscles are involved in producing movement. They need to know about disorders, so how diet can affect bone and muscle development. So, for example, talking about, for example, calcium and perhaps the role of vitamin D as well, and how they're important for bone health. Osteoporosis, so what causes it? What are the symptoms? How does it affect the bone strength? And as I've said, in terms of assessment, using label diagrams, giving explanations of movement and of the structure of joints and the function of the skeleton. So there are opportunities for using, applying knowledge as well as recalling knowledge and understanding as well. So moving on to topic 5. This is about coordination, and we're looking at nervous communication. We're looking at the endocrine system, hormonal systems. And we're looking at how these systems detect stimuli, how they respond to stimuli, how sort of balance is maintained within the body. and how behavior and health is affected if these systems aren't working properly. So in terms of the nervous system, students need to learn about the typical areas of the nervous system that we would see in a traditional biology specification. So they need to know about the structure of neurons, they need to know about the central nervous system, reflex arc, synapses and how nerve impulses cross synapses. They need to know about the regions of the brain and the roles of these regions. I can speak from, well, experience as an examiner that learning about the brain and the roles of regions of the brain is not something that students find very easy. We had a question in the 2025 paper about regions of the brain in the UK paper, and students found that quite difficult. But it's really a matter of learning, and I think that perhaps reflects that perhaps in the teaching, the students weren't focusing on learning the different regions of the brain, being able to identify where they are on a diagram, and they weren't that familiar with what the different parts of the brain actually do. Students also need to know about the structure of the eye and the ear. The eye is in the International GCSE biology specification, but the ear structure is not. Defects of the eye, again, in the I can speak from experience as an examiner that from the UK biology specification, learning about defects of the eye, understanding how they affect vision is not something that students find very easy. In terms of hormones, the students need to learn about some key glands such as the adrenal gland, the pancreas, and they need to know about specific hormones such as insulin, ADH, and adrenaline. And they need to be able to compare the nervous system with the hormonal system and hormonal coordination. So this again is an opportunity where students can be tested in terms of applying their knowledge and understanding of the nervous system and the hormonal system and be able to pinpoint any similarities and differences. So an interesting topic, but a topic that is not necessarily very easy in terms of getting to grips with the content. Continuing with topic 5, students need to learn about health and drugs, so the effects of alcohol, legal and illegal drugs and how they impact on the nervous system. They need to learn about mental health conditions and the symptoms and treatment of these conditions, such as depression and Alzheimer's disease. And looking at the focus on assessment, so giving definitions, learning the position structures within the body, such as the location of the endocrine glands, being able to identify structures of a neuron, being able to identify the components of a reflex arc, etc., etc. Really good opportunity for, particularly for visual learners, being able to pinpoint the structures, the names of the structures, the functions of the structures. And as I said earlier, if you do have any specific questions about any of the content, etc., just put your questions into chat and I will attempt to answer them. So topic 6 is about nutrition and about energy. And there are clearly some links with earlier topics. So, for example, topic 2, biological molecules, students had to learn about the structure of carbohydrates, etc. And here in this topic, they need to know about the importance of some of these biological molecules in the diet. And they need to know about how the sort of the needs of the body, the demands of the body change with age and activity, with climate even, and also and with pregnancy. So they would need to draw on some of the knowledge of the structure of these molecules as well. They need to know about deficiency diseases, such as the lack of vitamin C, the lack of iron, the lack of vitamin A, and the lack of protein in the diet. They would need to know about the symptoms of some of these deficiency diseases, such as the symptoms of scurvy and kwashiorkor, for example. The topic continues with learning about the digestive system. So the structure and function of the alimentary canal. They need to learn about peristalsis. So good opportunity there for just, little demonstrations to demonstrate the action, for example, of the muscles in the esophagus to bring about peristalsis. They need to know about the role of fiber in the diet as well. So again, in a previous topic, in terms of enzymes, students had to learn about factors affecting enzyme activity. They should have done some practical work involving enzymes, but there's opportunity here for doing practical work related to enzymes as well. So they need to know about where these enzymes are produced, what they break down, and they also need to learn about the role of bile and in the alimentary canal as well. So bile in terms of neutralizing acid from the stomach and emulsifying fats. So again, some interesting biology here, and some clear links back to work covered in previous topics. Obviously, as teachers, you are free to organize your teaching as you see fit. And we'll mention that later on when we have a look at schemes of work. But you might, for example, make a link with topic 6 and the previous topic that covered some of this, or previous topics that covered some of this work on carbohydrates, etc., and enzymes. So continuing with topic 6, students also need to learn about the absorption of the breakdown products of digestion. They need to learn about teeth. So in terms of absorption, we need to know about the adaptations of a villus, and we need to learn about the structure of teeth and how to look after our teeth and how teeth develop. And then moving on from there, we learn about health and food safety. So BMI calculations, possibly also thinking about waist to hip ratios. We need to think about the link between BMI and obesity. We need to think about our diet and the levels of cholesterol in the blood and perhaps how to reduce that. We need to consider diabetes. So again, links with our diet, diabetes, type 1 diabetes, type 2 diabetes. We need to also consider food hygiene, and we need to consider food storage. And in terms of assessing the content of this topic, so practical work with food tests, so knowing about what is the test for example, glucose. How do we know if the test shows that glucose is present in a food? So obviously we can use the same principles for testing for starch, testing for protein, testing for lipids. We could use label diagrams, for example, the structure of the alimentary canal. We need to learn methods, learn safety issues, perhaps related to these methods. So for example, for the Benedict's test for glucose, a practical consideration, a safety consideration would be heating up mixture of solutions and safety issues regarding that. We could have some data interpretation exercises So students could perhaps be presented with information about the results of a food test and be asked to interpret what that information tells them about the contents of the food. Likewise with BMI rather, they could be given some information about BMI, be asked to do a calculation, and then use the data from that information, that BMI calculation, to be able to think about, for example, the health risks or risks to health to a person. And also being able to give explanations about the outcome of sort of enzyme reactions as well. So again, interesting topic, very varied really, and lots of different opportunities to assess students. Topic 7 is related to respiration and obviously energy. So we're looking at the release of energy, the transfer of energy. Students need to be familiar with aerobic and anaerobic respiration. They need to know the word and symbol equations for aerobic respiration and just the word equation for anaerobic respiration. It's important as well, I'll just mention this now. So in terms of symbol equations, if, for example, a student is writing carbon dioxide in symbols, it's very important that the 2 in CO2 is subscript. Because if, for example, they put capital C, capital O, and then a superscript 2, then that's incorrect. That is not the correct symbol equation for carbon dioxide. So it's important to stress to students that when they're writing symbol equations, the symbols need to be correct. And students need to be able to compare aerobic and anaerobic respiration in terms of using or not using oxygen, in terms of how much energy is produced, and the products of these two types of respiration. And then they need to know the basics about ATP and the transference of energy using the phosphate groups, the additional or removal of phosphate groups. And in terms of practical work, students could compare the level of carbon dioxide in the air that we breathe in compared with air that's breathed out. So simple experiments, for example, using lime water would be okay there, possibly using bicarbonate indicator. So in terms of assessment, the use of equations, obviously learning the equations, particularly the symbol equation for aerobic respiration. Students have to be able to compare aerobic and anaerobic respiration. They need to perhaps be able to interpret data that has come from practical work. And they need to be able to give explanations. So for example, in terms of the function of ATP. So I've just noticed that we have a question in chat. So the question is, do they need to learn how to draw specific diagrams with labeling, or just need to know the proper labeling? So thank you for the question. The answer to the question is that we would not normally expect students to draw a diagram of a structure. We would certainly expect them to be able to identify structures on a diagram which they're given. So we would not perhaps expect, for example, a student to have to draw a diagram of a villus, for example, a diagram of an eye, a diagram of the alimentary canal, but we do expect them to be able to either perhaps add a labeling line to a diagram that they're given in a question. Sometimes we use multiple choice questions to assess knowledge of structures. So students would need to, for example, know the location of an endocrine gland within the body. So learning to draw specific diagrams, no. Learning the proper labeling, yes. So hopefully that's answered the question, and thank you for raising it. That was a good question. So moving on, so we've covered topic 7. As I said, there are 12 topics altogether. Topic 8 is about gas exchange. So we need to learn about the lungs. We need to learn about how exercise and lifestyle affect the respiratory system and also the cardiovascular system. So in terms of content, we're looking at the thorax and breathing. So we need to know about the lungs, structure of alveoli, the role of the diaphragm, the role of intercostal muscles, how they bring about ventilation. We need to look at the adaptations of the lungs for efficient gas exchange. So here we can bring in a link from earlier topic about the movement of substances into and out of cells. So we can bring in diffusion, and we can think about the structure of an alveolus, for example, how it's adapted for gas exchange, how it's adapted for efficient diffusion. Opportunity for practical work. We could measure tidal volume, we can measure vital capacity, we can use a spirometer, we can use very simple spirometers to measure tidal volume, etc. If you don't have access to simple spirometers, you can get information. You can use animations, for example, so that students understand what a spirometer is, and they can interpret information from a spirometer trace, such as measuring tidal volume or vital capacity, and breathing rate as well. We expect students to understand how exercise affects breathing and pulse rate, and we can also link in with that references to oxygen debt, and also references to the effect of adrenaline on heart rate or on pulse rate. And thinking back to sort of homeostasis and the regulation of systems in the body, students need to learn about chemoreceptors and the fact that they detect carbon dioxide, and that's involved in the control of breathing. And in terms of health, students need to know that, or know the beneficial effects of aerobic exercise, and they need to be familiar with the risks of smoking or smoking tobacco in particular. So I've already mentioned in terms of practical work, you can measure lung capacity. So when I was teaching, we used to have these very, very long, effectively long plastic bags, cylindrical bags with a mouthpiece, and you can use that to measure, get a basic idea of measuring lung capacity. So something quite simple, and obviously there are different methods for measuring breathing rate, different bits of equipment that you can use, or just simply sort of measure the movement of the thorax before and after exercise. So there are different opportunities here for assessment. It could be identifying structures on a diagram. It could be explaining the role of these structures, applying knowledge to perhaps slightly unfamiliar situations, but just to apply knowledge of how, for example, ventilation takes place in humans and applying that knowledge to a different situation. Being able to interpret data from different scenarios, such whether it's the use of a spirometer or whether it's data collected from exercise, such as data to do with pulse rates, for example. So topic 9 is to do with transport. So if you like some traditional biology here, So looking at the composition of the blood, looking at the circulatory system, the function of the heart, and how we can treat heart disease. The blood and transport, so we need to know about red cells and white blood cells, plasma and platelets. The fact that carbon dioxide is transported in the plasma, and also nutrients and urea and hormones, and the fact that the plasma and the blood distributes heat around the body. And they need to know about the formation of tissue fluid. Circulation in the heart, so we need to learn about the structure and function of the heart and vessels, and the route that blood takes to and from different organs in the body. Again, from personal experience as an examiner, I can say that some students are not very good at remembering the circulation of the blood into and out of the heart. So, for example, left ventricle pumping blood out into the aorta, then it goes around the body and then returns to the heart via the vena cava into the right atrium, and then it moves to the right ventricle and pumped to the lungs in the pulmonary artery, etc. I can't stress enough the importance of students learning sort of that basic information about the circulation of the blood. Students also need to know about blood pressure, so systolic and diastolic pressure. So again, you might choose to use a blood pressure monitor to measure these things, get some practical experience. In terms of the health aspect of topic 9, and you'll notice that in several of these topics now there's an aspect which focuses on health, which is obviously important. So we need to know about the causes of heart disease, how it can be prevented, what treatment can we give. So for example, the use of stents, the use of statins and beta blockers and ACE inhibitors, and also the use of monoclonal antibodies in diagnosing and treating cancer. So as with the other topics that we've considered so far, again, good opportunity to test knowledge and understanding using diagrams, questions about the function of different structures, and scenarios which have a real world background. So lots of information available about heart disease, for example, how it can be treated, how it can be prevented. So a good opportunity for perhaps introducing fairly up-to-date medical information to do with treatment, diagnosis, etc., of heart disease. Topic 10. So this is about homeostasis. So we need to think about temperature regulation, water balance, and the control of blood glucose concentration. So in terms of temperature, we're looking at the roles of the skin in terms of sweating. We need to know about vasodilation and vasoconstriction. We need to know about the role of shivering. Excretion, so we're thinking here about how is urea removed, also how is it formed, what is it formed from, where is it formed. The removal of carbon dioxide, so previous topic we've looked at the lungs, so removal of CO2 from the lungs. Water balance, so we need to know here about the role of the kidney in water balance, and also the role of ADH in water balance or osmoregulation. Glucose, so diabetes was considered again in a previous topic, but here we're looking specifically at how blood glucose concentration is controlled through the action of the hormones insulin and glucagon. We need to know about the role of the hypothalamus, and we need to know about negative feedback. So negative feedback, again, is a more challenging aspect of this part of the topic. And then in terms of the liver, so producing bile, producing urea, its role in detoxification, so removing toxins from the body, and also its role in glucose regulation in terms of storing glycogen. And again, in terms of health, so kidney disease, treatment of those diseases through dialysis, kidney transplants, comparison of the pros and cons of dialysis versus kidney transplants. So as with previous topics, again, some health issues here. And in terms of practical work, you could use visking tubing to model situations, for example, thinking about what happens in the kidney, and visking tubing could be used to model situations. But equally, visking tubing is used, as I'm sure you know, in practical work that we do regarding, for example, enzymes and the breakdown of substances and the diffusion of small molecules through the visking tubing. Visking tubing, for those of you not aware, is a tubing which is partially permeable. And again, as with the other topics, again, there's opportunities for assessing knowledge and understanding using diagrams, and sort of asking students to explain particular processes. So for example, just going back to the previous slide, negative feedback, the role of the hormones, etc., in terms of in controlling glucose concentrations in the blood. Good opportunity for using diagrams to explain that particular process. Topic. I'm just going to skip to the next slide. Okay, so I've not missed anything out. So topic 11, reproduction and heredity. So we got, again, a wide range of coverage here. We've got human reproduction, we've got contraception, we've got inheritance, we've got genetic variation. So in terms of reproduction, students need to know that there is, fertilization results in the formation of a zygote, which then develops into an embryo. They need to know about meiosis and the production of haploid gametes, and the fact that meiosis results in variation in organisms. They need to know about the basics of the reproductive systems. They need to know about the menstrual cycle, the role of hormones in the menstrual cycle, and they need to know about the structures that are involved in pregnancy, so the placenta, for example. They need to learn about methods of contraception, so hormonal methods, barrier methods, natural methods, IUDs, and sterilization. So consideration of pros and cons of these methods of contraception, and they need to learn about in vitro fertilization as a method of treatment, and perhaps consider the pros and cons of IVF, such as, for example, certainly in the UK, one big drawback is the cost of IVF. Can be very expensive. And then we need to think about inheritance and genetics. So here we've got some different terms been introduced with what students might consider to be relatively complicated names, so alleles, genotype, phenotype, dominance, codominance, etc. So there's sort of new vocabulary here. Students need to understand what these terms actually mean. They need to be able to interpret information, for example, interpret monohybrid crosses, be able to sort of draw diagrams or complete Punnett squares, for example, to show monohybrid crosses, or to explain sex determination. And then we've got sort of the health considerations, so genetic disorders and gene therapy to deal with some of these genetic disorders. So again, that we can introduce some up-to-date sort of medical science in terms of gene therapy here, for example. And then the final aspect of the content of topic 11 is variation. So we need to think about variation caused by genetics, variation caused by the environment, or variation caused by both of those factors. And linking with a previous aspect of the topic, just considering that random fertilization increases diversity, as does meiosis. Assessment. So I mentioned interpreting monohybrid crosses, for example. So students need to be familiar with genetic diagrams and sort of solving inheritance problems and giving explanations of variation. Okay, the final topic of the specification, topic 12. So we've got here a focus on disease transmission, immunity, treatment of diseases, and useful microbes. So in terms of disease, we need to think about the fact that there are different stages of infection. So we've got infection, then we've got incubation period, then we get symptoms. And students need to know that diseases can be caused by different types of pathogens, so viruses, bacteria, fungi, and protists. So protists such as malaria, for example. Transmission and prevention. So we're looking at some different diseases here. So HIV, Ebola, cholera, gonorrhea, athlete's foot, malaria, and typhoid. The students need to know what type of pathogen causes each of these diseases. How can we prevent the transmission of the diseases? So here we can consider vectors which are involved in some of the diseases. So for example, mosquitoes in transmitting malaria. How can we prevent the transmission of the diseases? How can we treat the diseases? In terms of specific microorganisms, the students need to learn about the structure of a virus, the structure of a bacterium. They need to know about how these organisms multiply, how they grow, and the effect of antibiotics on bacteria. So in terms of practical work, again, I understand in UK schools, practical work involving microbes can be very difficult because schools are not necessarily set up for being able to produce, for example, growth media, which is sterile, etc., etc. So practical work involving microbes can prove to be a challenge. I appreciate that. So again, this is where using perhaps videos, animations could be used if you don't have the first-hand opportunity to do this kind of practical work in your schools. But we'll talk more about practical work as we go along. Okay, continuing and finishing topic 12. So immunity. So again, this is an area of biology that students often find difficult understanding about antibody-antigen reactions, understanding different types of immunity, so natural immunity, artificial immunity, natural and passive, active and passive immunity. They need to know about antibiotics. So antibiotics was just in the previous section. but they need to know about the use of antibiotics and antibiotic resistance. So antibiotic resistant organisms such as MRSA. They need to know about the usefulness of microbes. So there's been focus on, the negative side of microbes causing disease, but also students need to know that microbes can be very useful. So involved in decay, for example, using sewage treatment and aerobic and anaerobic action. So again, here we can reinforce knowledge and understanding of respiration. So a good opportunity for making links between topics. And in terms of assessment, so case studies, for example, involving different diseases, comparing the action of different antibiotics, for example, and their effectiveness on different organisms. So looking at assessment here, there's a very, very good opportunity for applying knowledge and understanding to unfamiliar situations. So, and as we know, students find that difficult. So topic 12 can be quite in terms of the assessment can be quite applied. So not necessarily straightforward recall, learning facts and remembering those facts, but also applying knowledge and understanding of different situations. Okay, so that's quite a lengthy chunk of information about the content of the course. So as you can see, there's a lot of detail there. And so when it comes to planning your course, whether it's over 1 year or 2 years, there's a lot of information to cram in, core practicals to complete. So it's requires some careful thought when it comes to designing your course. Okay, so in terms of the topic content that I've just been talking about, any comments from you about topics that your students do or might find challenging? What kind of methods have you used to help students in the past? You've got any examples of what you've done to help students? So just an opportunity for you to reflect for a couple of minutes. And if you have any comments, put them into chat. So I'll just pause talking for a couple of minutes and then I'll respond to any comments that pop up. Okay, I'm going to continue. Okay, so yeah, I mean, very valid comment. As it's might be the same for several of you. If it's your first time sort of teaching this, you won't have any answers because you haven't got that experience. But I think as I went through those different topics, I probably perhaps highlighted some of the areas that students do find challenging and why they find them challenging. Yeah, and we've got another comment about homeostasis being tricky. And I totally agree. Um, some of the more abstract ideas do tend to be more difficult for students to grasp. Um, so I think we can probably move on now. But as I've already said several times, any questions, if you think about them, put them into chat and I will have a look as we go along. Okay, so we're going to have a look at the assessment structure. So there are two exam papers. They are untiered. So in other words, the papers they're graded from 9 to 1. So they're accessible to students across the full ability range. Just to put that into context, in the UK, we have tiered papers. So we have a foundation tier and we have a higher tier paper. So the higher tier is from grade 9 down to, in fact, grade 4, really targeted grade 9 to 5. And then the foundation tier is targeted from grade 5 down to grade 1. There are some questions that overlap in the higher and the foundation tiers, but we do have tiered papers, which perhaps you are aware of. But the papers for this specification are untiered, so they're graded from 9 to 1, and the opportunity for sitting these exams twice per year, November or June. And both papers must be sat in the same exam series. So, for example, 2025, 2026, etc. There is no practical exam. Practical skills are assessed in the written papers. And there are similar question styles in both papers. So the question styles are multiple choice, for example, short answer. Short answer will be between 1 and 3 marks. Then long answer, typically 4, 5, or 6 marks. We have questions which involve the use of data, interpreting data, and we have questions that involve planning. So they would relate in particular to practical skills. In terms of the paper structure, so paper 1, paper 2, each contribute 50% of the International Human Biology GCSE. Both papers cover the same 12 topics. Both papers are available twice per year, November and June. And both papers are the same length of time, so 1 hour and 45 minutes. Both have a total number of marks of 90. Both contain the same types of question, except there's a slightly different approach in paper 2 that we'll see in a moment. And in both papers, students can use a calculator. So just to summarize that, so both papers or each paper is worth 50% of the final grade, assessed by Pearson externally, available twice per year, 1 hour 45 minutes. Both cover the same 12 topics, and questions may come from any topic. So question styles we've talked about. So we'll move on from there. Okay, so in terms of the design of the paper and the design of questions, there is a progression of challenge. So the earlier questions tend to be more accessible, less challenging, and then as we get towards the end of the paper, there is greater challenge, and there's greater challenge, and some of the later questions are targeted at higher grades. So the idea is that students get some confidence in the early parts of the paper. There's things that they're able to do without too much of a problem. And then you get more demanding aspects as we move through the paper. But also within an individual question, you get what we call ramping. So you get a sort of a build-up of difficulty, ramping of difficulty. So you might start with a very straightforward bit of recall in a question, and then the question develops into some application and some evaluation. So it becomes more challenging. And doing that means that we can assess across the full range of ability. Right, so question regarding the sort of teaching, is it better to teach GCSE biology first and then start human biology, or teach alongside? Now, my answer to that would be, it depends on your situation in school, how your school sort of delivers the different courses that you teach. So, do you have the time to teach one and then teach the other? Do you have the time to teach them side by side? So, who would you target human biology at? Which type of student would you target the subject at? So I think to answer that question depends on your individual situation. I guess having general knowledge of biology can help with learning about human biology, but equally, there's not necessarily a need to do the ordinary GCSE biology first and then do the human. So I think it depends on your individual situations. Right, so there is a difference with paper 2 compared with paper 1. So it starts with a passage of information, a comprehension style task, as it were. And the whole point of that is to introduce some of the specification detail in context. So there's a passive information, and it sets up some sort of real world scenario. And then there are questions that follow that passage of information. And the idea is that perhaps 1 or 2 points can be drawn from that information to answer questions, but students have to use their own knowledge and understanding to answer the questions that follow the passage. So the passage puts things into context, but students have to use their own knowledge and understanding to answer subsequent questions. Questions might be synoptic, and that means that the answers involve making links between different topics. So, for example, knowledge about respiration, and then knowledge about the use of microbes and sort of respiration there. So making connections between different topics, enzymes and digestion, enzyme structure, enzyme action, enzymes in digestion, that would be another perhaps aspect of synopsis. And it's important that students are prepared for dealing with unfamiliar contexts. So the more that they're exposed to examples of questions, the better. Here is an example of what we're talking about regarding paper 2. So we have a passage of information. This happens to be about the heart and the blood. And then we have questions that follow. So students have to use their knowledge, draw on their knowledge. So main tissue, so they need to know that we're talking about cardiac muscle. Double pump, so they need to know about the circulation of the blood, etc. Then a multiple choice question about cell types in the blood, etc., etc. So the passage of information puts some of the specification content into a context, and then we have questions that follow, which have to be answered using knowledge and understanding. The very, very last question is a calculation, and there's an instruction to give answers in standard form. That's something I will mention a little bit later on. Okay, I'm not sure if you have any questions about that, but if you do, just pop them into chat. Otherwise, I will move on. So I'll just pause on this screen for a moment. If you have any questions about the papers, put them into chat. Otherwise, I'll move on. There are lots of past paper questions that you can access through the Pearson website. I'll just pause for another 30 seconds. Okay, so we have a question. Can I explain the practical part online solely, or will that mislead the students? If I guess from the question that you're perhaps suggesting that you don't have the facilities, the opportunity for students to do practical work firsthand. Is that correct? But if students do not have the opportunity to do practical work firsthand themselves, teacher demonstrations are an alternative, and obviously using videos and animations. It's important that students are able to, even if they're not able to do certain types of practical work themselves, it's important that they can at least visualize the way that different practical activities are carried out. So they need to know about the types of equipment used, how is the equipment used, etc., etc. So if you don't have first-hand opportunities, then other methods, like I said, using a demonstration, using videos, etc. So, okay, so looking back in the chat, yeah, so like I said, so if going to school isn't available in a region, then yes, using, if you're if you have facilities from wherever you're doing your teaching from to demonstrate equipment, but use videos, pictures, diagrams. You can show diagrams of how equipment's set up and explain it, etc., etc. So any way that you are able to show students how equipment is used, how measurements can be taken using that equipment, etc., then that's fine, as long as students do have that opportunity to at least be able to visualize how a practical is carried out. So thank you for the question. Okay, so the assessment objectives. Students need to know about, well, in terms of the papers, then they're not going to be able to know if a question is AO1, AO2, AO3. It's not actually explicit, but if a question is asking for them to recall some knowledge and understanding, then it's AO1. AO2 is applying knowledge and understanding, and also some analysis. So, for example, some calculations are AO2, and then AO3 is largely focused on experimental skills, analyzing data, evaluating data, evaluating methods. And you'll see from the breakdown of marks that AO1 and AO2 have an equal weighting of between 38 and 42%, and AO3 is half of that, between 19 and 21%. So the assessment objectives, we use those when we're writing papers, and we have to stick to those proportions of marks. So between 38 and 42% for AO1, recalling facts, definitions, concepts. AO2, being able to apply understanding to unfamiliar contexts, to be able to interpret data. AO3 is focusing on practical work, so experimental skills, analyzing data, drawing conclusions, evaluating what evidence has been collected, evaluating methods, how could a method be improved, and the one individual question might cover all three assessment objectives. So it could involve some recall, it could involve a little bit of application, and then some work involving experimental skills. Okay. So AO1, we've already said it's about recall. Students like AO1 questions because if they're a kind of student who is good at learning facts and so on, then they'll be able to answer AO1 questions without too much problem. But AO1 on its own is not sufficient to get a top grade. So having a good knowledge and understanding of facts is not sufficient on its own to get a good top grade. You do need to be able to apply facts, you need to be able to analyze and so on. So typical AO1 question here, using recall. So students have got to put the appropriate term or terms into the box. It's about knowing about what's involved with regulating water levels in the blood, what's involved in transporting oxygen, and so on. So choose the correct word from the box. And then the question on the right about the eye, it's a matter of, okay, I need to learn the parts of the eye. So learning that part W is the pupil and part X is the retina. And then function of part Y. So part Y is the optic nerve. So they need to be able to give the function. So typical AO1 questions. So different styles of question, could be multiple choice as well. AO2 is this application of knowledge, analysis, evaluation. So using biological knowledge in familiar and unfamiliar situations. It could be involved graph work, analyzing a graph, doing a calculation. We're often using command words such as explain, suggest, or evaluate. So slightly more challenging command words in some respect. But whatever the situation, the biology required to answer the question is always from the specification. Often AO2 questions require some sort of reasoning involving different steps. So for example, a student might come out of an exam and said, oh, we didn't learn about enzymes in milk. Well, yeah, that's correct. There's nothing specifically about enzymes in milk in the specification, but students can use their knowledge and understanding of enzymes to answer questions about enzymes in milk. We never ever write a question that student can't access. All questions have to be linked to specification points. So modeling, etc., those kind of ideas can be used to help students learn about how to respond to AO2 type questions. Obviously, the more practice, the better. The more practice at answering questions which have some sort of unfamiliar context, that will help them to develop their skills here. So it's all about thinking as a scientist and not just recalling facts. So here we've got a question about edema. Question tells the students that too much water enters body cells. So question is, explain how water can enter body cells. So all the students have to do here is use their knowledge and understanding of osmosis, movement of water. And so the situation is unfamiliar. They've never been taught about edema, but they have been taught about osmosis and the movement of water. So that's all they need to do to answer the question here. And then here we've only got part of the question, but it's an explanation. So it obviously involves data. Use data from the table to help your answer. So students have to explain why males need a different amount of energy compared with a female in that particular age range. So we don't have the data, that doesn't matter. But AO2 can involve interpreting data, giving an explanation, using knowledge. So they would need to use knowledge of the fact that in general males require more energy than females. Then AO3, also some analysis, but we're evaluating methods, we're evaluating data, evaluating experimental design, but also students need to be able to plan investigations, need to be able to write plans. So in terms of the practical skills, we're looking at understanding of variables, using a control, looking at data, being able to draw conclusions. So a question might often focus on, how could you improve this investigation? What are the limitations of this particular method, etc. So the questions may have command words that are more demanding. So evaluate is a more demanding command word, requires sort of a higher level of cognitive skill. Justify, discuss. So these are sort of higher level command words. I'm sure that some of your students have the use of variables, etc., drilled into them pre-GCSE, but they need to know about independent variables and dependent variables, how variables are controlled. They need to be able to spot errors in data, suggest how data quality could be improved. They need to be able to pick out anomalies and perhaps think about, well, why could an anomaly occur? So AO3 requires students to actually think like a scientist, and to be able to look at a situation and give some explanation of what's been going on. So a couple of typical AO3 questions. So, okay, we don't know the context of the investigation, but name the independent variable. Give two variables that should be controlled. So students look at the situation that they've got, think about, okay, what needs to be controlled here? And then state how the student could improve reliability. What could they do? The command words here are all quite straightforward. Name, give, state. The next question is about using some equipment to compare the energy content of different foods. You notice this has a mark allocation of 6 marks. So students have to effectively describe a method here. So there would be perhaps 12 answer lines, and they have to give a method, which includes some detail about how the equipment shown could be used to compare the energy content of foods. Okay, so command words, question types. We've already said there are different styles of question. So multiple choice questions can be used to assess all 3 assessment objectives. Short answer questions typically use the command words sort of just describe and suggest and explain and perhaps state or give or name. So easier, less demanding command words. Longer answer questions, which are typically between 4 and 6 marks, they use, in general, more demanding command words, such as explain, evaluate, etc. And obviously the longer answer questions require perhaps responses which are more structured. Several points need to be linked together. So in terms of planning questions, we just saw the one to do with energy and food. They focus on methods, variables, controls, and they're testing knowledge and understanding of practicals, knowledge and understanding of the core practicals. Maths question, so there could be a calculation, calculate BMI, for example, draw a graph of some data, plot the points, label axes, scale the axes, look at data, look at graphs, analyze that information. And then we've got the paper 2 question, the comprehension. So we've got the scenario that is looking at making links between different topics, so synoptic understanding, and using questions that apply knowledge across several topics. So that's the synoptic understanding. So it's important that, from the get-go of the course, you expose your students to different styles of question. As you go through the course, don't sort of save the more difficult types of question till later on towards the end. Try to introduce students to different question types as you go through, especially the ones that are not just recall, so the AO2 and the AO3 questions. Try to get the students exposed to different question styles early on so that they become familiar with them, so they become more confident with them. So multiple choice questions, they're not necessarily the easiest ones. As we said, they can assess all three types of assessment objective. It's not just about memorizing facts. And a multiple choice question could involve interpreting data or even applying knowledge. So of the total number of marks, so total number of marks is 180 for the two papers. About 15% is pure recall. And success here is about being able to use data, being able to think critically, being able to look at the distractors. So we have 4 distractors in a multiple choice question. Students need to be able to think about the question, look at the distractors and think, okay, what can I eliminate? What does that leave me? So some distractors can be eliminated fairly easily, others less so. So students have to be able to look at the distractors. And often when we see examination papers that have been completed, we see distractors that have been crossed through because the students have looked at them and they've eliminated them, just leaving the one that is what they consider to be the correct answer. So you can use multiple choice questions frequently, good for sort of checking knowledge and understanding, also good for trying to identify misconceptions. So they're good for quick assessments. So we've got multiple choice question at the bottom here about the brain. So which part of the brain controls sweating? We have a different kind of question above that, the draw one straight line. So that's just making links between a structure and a function or a feature. Typical question that checks recall. And the question on the right, so the fill the gap type of question. So we have a box of words. Students have to, in this case, choose, 5 words to complete the passage. And again, that's using recall of knowledge. So we can use multiple choice to test recall, but we can also use other methods, such as the draw one straight line from one box to another and use words from the box to fill in the gaps. So short answer questions, they require concise answers. It might even be one word answer, typically used to assess AO1 and AO2. Students do not need to write in long sentences, just short points, bullet points are fine. And if a question is worth, say, 3 marks, you need to guide students to make 3 points. And if it's 2 marks, they need to give 2 points. So here we've got a couple of examples. Describe the function. So this is largely recall, function of red blood cells, 3 marks. So they give 3 points. They could write 3 bullet points. That will be fine. So the next bit, state the type of blood vessel shown. So students are shown a photograph. They need to recall what kind of blood vessel has been shown, and then explain your choice. So here they would have to give reasons why they have chosen whatever type of blood vessel it is. And then the longer answer questions. So between 4 and 6 marks. Mark schemes are points based. What that means is that it's effectively 1 mark is given per valid point written. Students do not need to write in full sentences. So again, they could use bullet points if they choose to do that. I would always advise that short sentences end up giving better structure, end up with the examiner being able to decipher what the students are trying to say, much better than long, convoluted sentences. Level-based mark schemes are used, for example, they're actually used in the UK GCSE sciences, and they're also used in international A-level biology as well. But here all the mark schemes are what we call points based. So 1 point gives 1 mark. Language needs to be precise, it needs to be accurate, it needs, of course, to be scientific. And as I've said, bullet points are okay. And students should always look at how many marks are being awarded for the question, not worry about how many lines there are, how many marks are there, how many points do I have to give. Spelling, we know that many students are not brilliant at spelling. We will accept phonetic spellings. So that is only unless a word could be confused with another. So the example we've got here is mitosis with an E after the M. Because that we're not sure there is a student trying to tell us it's mitosis, or are they trying to tell us it's meiosis? We don't know. So we would not accept that. But other words, I know that we're talking about human biology here, but a good example would be photosynthesis. So for example, if a student wrote photo, F-O-T-O, and then synthesis, that's fine for photosynthesis. But the example we've got here is very specific, and we would not accept that. So we've got 2 examples of longer answer questions, one worth 4 marks, one worth 5 marks. So one question about tooth decay and the other about ADH in osmoregulation. And just to say that the osmoregulation questions like that, sometimes it might help students, and sometimes students do this. They might draw a very quick sort of flow diagram, which they use then they might annotate it and then use that to draw ideas from to answer the question. So using simple diagrams in the answer is perfectly acceptable if it helps the students. And obviously, if they've got a diagram and of a process and they label it and so on, you can also obtain marks from that. So a couple of typical longer answer questions. The experimental planning questions, which come under the umbrella of AO3. So students might be planning an experiment in an unfamiliar context. So they will have done core practicals, and all students have to do here is to just apply the kind of principles they've learnt for one core practical and apply it to an unfamiliar situation. Some of you might be familiar with CORMS, which is a framework we use in international GCSE biology in terms of practical work. It works very effectively. It's a very good framework for planning practical work, planning investigations. So CORMS is just an acronym. So we have C for change. What are you changing in the investigation? Students need to be specific about what they're setting up. We have to identify organisms. It could be the organism could actually be an enzyme, for example. They need to be specific and talk about measurable things. So, same species of fish, same enzyme. Everything's needs to be specific and not generalized. So the R in CORMS is for repeat. So students need to talk about repeating values, repeating measurements. Why do they do that? So, for example, calculating in a mean. They need to write about what will be measured at the beginning of the investigation, after the investigation has taken place. Are they measuring length? Are they measuring mass, etc.? They might write about time intervals. How frequently would they take a measurement? How will they take a measurement? Will they use a millimeter ruler? Will they use a measuring cylinder? Will they use a gas syringe? That kind of thing. They need to talk about what they're keeping the same. So usually two relevant variables, whether that's temperature, light intensity, pH, using vague terms such as, I would keep the conditions the same. That is not advised. They need to be specific about the variables that should be controlled. So it's best to, what we sometimes see is that students actually write that acronym CORMS, and they will, next to each letter, they will write briefly what they are going to change, what they're going to repeat, etc., etc. But the advice is to perhaps do that and then write some steps. It could be numbered steps. Number 1, do this. Number 2, do that. Just short statements about the different parts of the method. But CORMS is a good framework. So if you're not familiar with it, these slides give you an idea. It's very useful. So here we have an example. We've got a question to do with enzymes. It's to do with the activity of an enzyme and temperature. So the question is, design a method the student could use to obtain the results shown in the graph. So we've got some points which have come from a mark scheme about putting a test tube substrate in a test tube, put it in a water bath to control temperature. So water baths could be different temperatures, using a substrate, mixing it with the enzyme, measuring how long it takes for a reaction to occur, whether that's a color change or, for example, how much glucose is produced, keeping things the same, so the same volume or concentration of the enzyme, keeping the same concentration of substrate, using different temperatures. Words like amount we do not like. We want specific things such as volume or mass, concentration. So a mark scheme will have a list of points that we would expect to see in the student's answer, typically worth 6 marks. So this is just a little extract of a question and an extract of the mark scheme. Just gives you an idea of the kind of situations that we talk about. Sorry, I've just skipped a slide. And another example to do with pulse rate. So student investigates the effect of time spent exercising. So the question is, describe how the student could carry out the investigation. So we would expect students to have some personal experience of or knowledge of measuring pulse rates and how pulse rate links to exercise. So the mark scheme extract at the bottom. So students would need to write about taking the pulse before exercise and after exercise. How will they take the measurement? So, measuring in the wrist, measuring in the neck, using a pulse meter, that kind of thing. So some kind of electronic device, some kind of smart watch. Do some exercise. How long will the exercise for? That kind of thing. Measure the pulse rate after exercise. Allow some recovery time for the pulse rate to return to normal. Repeat the exercise. So we've got the repetition there. Controlling variables. So what you're going to keep the same, etc. Same person, same gender, same type of exercise, exercise for the same period of time, exercise with the same intensity, keep the environmental conditions the same, etc. So here we can see there are 7 points in the mark scheme, but there could easily be, this isn't the complete thing. So there might be, for example, perhaps 10 or 12 points, and the students have to get 6 points to score full marks. So this is a good example of a points-based mark scheme. 6 points, 6 marks. But if you use that CORMS framework, students can focus on the kind of things that they should be including in an answer. Okay, so we've already said a lot about command words, mentioned them a lot. So sometimes students do confuse words like describe and explain, but there is an appendix in the specification which covers these command words, and I'll show you that in just a moment. Here we go. I don't expect to be able to read all of those, but sometimes teachers share this sort of glossary of terms with the students. Some of them speak for themselves. So looking at describe, very typical command word, give an account of something. It's a matter of basically stating facts. No reasoning has to be given. But then if you move down and look at explain, an explanation requires a justification or exemplification of a point. The answer must contain some element of reasoning or justification, which could be mathematical. So an explain question might begin with a statement of fact, but then there has to be some sort of reasoning. So this happens because, that kind of idea. So some of these command words are not used very often in questions. Some are more demanding, such as justify, such as evaluate. Some are very self-explanatory, such as calculate, such as draw, such as label. So, like I said, often teachers do share this glossary of terms with their students. And as we've already said, there is sort of a hierarchy of cognitive demand. So think words like state and name and describe are have lower cognitive demand than command words such as evaluate and discuss. Okay, I mean, if you have any points here, then please do share them in chat. But obviously, you're going to introduce these command words, particularly through testing students' knowledge and understanding of the topics with exam questions. Using exam questions little and often to check knowledge and understanding is good practice. And in doing so, you are introducing command words and obviously ensuring that students are familiar with them and not fazed by them when they meet them. Okay, so we've talked a fair bit about practicals. We'll just have a further sort of delve into this aspect of the course. So, as we've said, practical work is embedded throughout the course. In the specification, there are italicized points which identify the core practicals. So this is about developing scientific knowledge and also developing hands-on investigative skills. But I think from one of our colleagues who is with us on the meeting, those hands-on skills are not necessarily easy to develop, easy to access. So we appreciate that some students learn their sort of scientific knowledge, their practical knowledge from watching videos, watching demonstrations, which obviously is absolutely fine. The practical skills, as we've already said, they're assessed within written papers. There is no written exam. And AO3 is roughly 19 to 21% of the exam marks. So the practical skills, AO3 skills, roughly 20% on average. We do know that students, if they're able to carry out a practical themselves, they have a huge advantage because they've done it, so they recall information from a particular practical. But equally, as we just said, some students are just not able to have that experience because of the situation that they're in and the availability of resources. So here are some of the core practicals which are in the specification. As I said, in the specification, they are in italics. So we've got sort of measuring vitamin C content, energy in food, effective temperature and pH on enzymes, immobilized enzymes. So that could involve, for example, the application of the use of enzymes, removing lactose from milk, sensory receptors in the skin. So using sort of pins to check sensitivity, for example, using or measuring the range of frequencies that we can hear, looking at how much carbon dioxide, this would be a qualitative experience, but looking at how much CO2 is in the air that we inspire and expire, effective exercise on breathing rate and lung capacity, exercise on pulse rate, and then diffusion. So the diffusion of substances across a partially permeable membrane, such as an artificial membrane like Visking tubing. So these are from different topics. So no coursework, no practical endorsement. So, for example, in the UK GCSE science specifications, the head of centre has to sign a declaration that practicals have been completed during the course. For the international GCSEs sciences, that is not a requirement. We've already said core practicals are assessed with written exams. There are, in addition to the core practicals, there are some other practicals that are sort of identified in the specification. And any apparatus and techniques, they align with the UK GCSE science specifications as well. And practical questions could appear in AO1, AO2, or AO3 questions. So if it's a simple recall question about a particular practical, then that could be AO1. It could be assessed through multiple choice, for example. So in terms of what students are assessed on in this practical context, they've been asked to solve problems, apply what they know about experimental methods and so on, do some planning, select an appropriate technique, describe methods, think about safety, think about variables, be able to record results methodically, look at data, analyze it, interpret it, draw conclusions, be able to use language correctly, correct technical language, names, pieces of equipment, and so on, and be able to think about accuracy, reliability, and validity in terms of sort of data evidence, and also then think about methods and identify improvements. We have a couple of questions in chat. So the UK exam paper different to international? There are differences in the two papers, but in the UK papers, the practical skills are still assessed in the 2 written papers. So GCSE biology in the UK, the practical skills are assessed in the same way that they're assessed in the international papers. So there's no, again, in the UK GCSE sciences, there's no practical exam. We do have to, like I say, we have to give this endorsement that the practical work has been carried out, but there's no separate exam. So the way that the practical work is assessed in the UK papers is the same really as it is in the international papers. And another question about the ratio of AO1, AO2, and AO3. So the ratio is, well, we've looked at the proportions already, but the ratio is between sort of 39, 41% AO1, 39 to 41% AO2, and 19 to 21% AO3. So that's always the same, and it's the same on the UK papers as well. So there are some distinct similarities. One difference which I've alluded to is that on the UK GCSE sciences, we do have a small number of questions which are marked using level-based mark scheme rather than a points-based mark scheme. So a couple of questions here related to practical work. So we've got some information shown, and we've got the question is, draw a diagram to show how the equipment is set up. So this will be equipment used to show the difference between carbon dioxide in inspired and expired air. So students will be expected to be familiar with how this simple equipment is set up and then just do a simple drawing of it. And we have another question which is to do with the effect of temperatures on receptors in the skin. So we're given part of the method, putting the hand in hot water, etc. They have to identify something to do with safety. So state one possible hazard with the method. So that would perhaps be to do with, making sure that the water isn't boiling hot, otherwise they would be scalded. And then how to reduce the risk of this hazard. So for example, perhaps using a thermometer to measure the temperature before hands are actually placed in it. So that will be typically, but so this shows you, that students need to be considering safety aspects of practical work as well as the mechanics of actually carrying out a practical. So it's important, even though you don't have to make a declaration, it's important that the core practical is completed because they are an integral part of the specification. You can obviously fit the practicals into your general teaching, or you can have some dedicated sessions, some standalone sessions where you do practical work. We're looking at obviously students knowing about the methods, making observations, recording results, and then analysing what they've got. Students don't have to use a separate lab book. They can just use sort of file paper, exercise books, whatever they would normally use. And obviously, if you are able to do practical work, you can use sort of a carousel approach where students move from one station to another to do perhaps a slightly different activity. Students can work in groups. It's just important that they all gain experience of what's going on. And as I've already said, teachers can give demonstrations, which could be recorded and then shared with students afterwards. And there are also, again, as I've already said, within the specification, there are some suggested additional practicals. They do not have to be completed, but if you do have the time and the facilities, then it just gives students a broader experience of practicals within the course. But for many of you, that could be the time constraints could be an issue. Here we've got some of those additional practicals. They're in an appendix in the specification. So we're looking at, the use of a microscope, looking at stages of mitosis, looking at nutrient content of foods, et cetera, et cetera. So it's just some additional practicals which help to broaden students' experience. But like I said, I appreciate that you may not have the time or the facilities to actually carry some of those practicals out. But practicals, it's important to think about, how you use them within the scheme of work. It's important that they do reinforce theory, so the two things go hand in hand. And as you're going through, it's important to be clear about, what are you measuring, why are you measuring it, which variables need to be controlled, how do you control them, and what makes the results valid, what makes the results reliable. And even if students collect data and they think it's not good, or that they often say, oh, this practical didn't work very well. Any data collected can be evaluated. So even what you might consider to be poor data is still useful in terms of being able to think about, okay, well, what improvements could we make if we want to improve the quality of the data? So good evaluation opportunities. There are some guides available for international GCSE sciences, practical guides. So you can see there's a specific one for human biology. These are free to download from the Pearson website. So you can see we've got a specific core practical guide for human biology. And then there is a guide on using practical terminology, which covers all of the international GCSE sciences. Okay, so I think we've already talked a lot about what we can do to prepare students for practical skills, etc. So we can probably move on from this. We're going to, and again, if you've got any questions, please put them into chat. But we've talked quite extensively about practical work and assessment, so I'm sure that we can probably move on. Like I said, if you do have any questions, please put them into chat. We're going to look at math skills next. Again, we've already talked about these a little bit. So just as with the other GCSE sciences, math skills are important. The assessment of maths, we are looking at using maths in context, not just standalone maths questions. They're in context with the content of the course. Altogether, we're looking at about 10% of marks, which are maths related. Those maths marks could be in areas such as data analysis, calculations, but also graphs and evaluating patterns. So it's not simply a maths mark is for doing a calculation. It could also be work to do with graphs. And maths questions, if it's a calculation, for example, that might be assessed under AO2, but evaluation, evaluating patterns and so on, that could be under AO3. So maths marks can be assessed under two assessment objectives. Okay, we have a question in chat, which I'll come back to in a little while. Okay, so in the specification, there is some detail about the skills required, but basically students need to be familiar with percentages, ratios, averages, estimates, standard form. If you remember earlier on, there was a question that was to do with standard form. So what's some a more demanding maths question might ask a student to give an answer in standard form, or to, for example, a particular number of significant figures. Some questions ask students to draw a graph and others to rearrange questions. So some of these skills are more demanding than others. So as I said, for example, giving an answer in standard form. But if you expose students to different types of maths skills in questions, in lessons, in context with the teaching, then it should build their confidence. And doing practice means that come an exam, hopefully the students will will be able to sort of answer questions confidently. And also in teaching prior to GCSE, so in Key Stage 3, if you can introduce maths in science, then again, they're going to become more confident. And you can use math skills in lesson starters. You can use math skills in exit tickets at the end of a lesson, where you perhaps just give a short, small piece of paper with a question on it, which involves a math question. They answer the question before they leave the room. Or it could be some sort of data handling task that has a mathematical content to it. So using and exposing students to math skills regularly is important, and it does make them more confident. So this is just an extract from the specification, identifying where in sort of biology, chemistry and physics, different math skills are assessed. So that's something that you'll see in the back of the specification. And here we've got some typical maths questions. So students are given some data. The first one, top left, they're asked to calculate extra mass. The one below that, they're asked to give an answer to 3 significant figures. And then we have one top right where students have to calculate percentage. And then the one bottom right, students have to calculate a ratio. So you can see that there are different types of question. The one bottom right, we have data that's given in standard form, which makes it a bit more complex for some students. So we've got a range of maths questions which could be used in an exam paper. And we have a guidebook. So I know this doesn't say on the cover human biology, but it does cover human biology. So another free resource that you can download from the Pearson website, so that you yourself and your students can be given additional guidance on the use of maths in science. So the kind of thing that we expect students to be able to do, some examples. So just coming back to the question that's in chat. So MM, if you could give me a little bit more detail about what you actually mean, then I might be able to give an answer. Thank you. Okay, so again, I think we can probably move beyond the current slide. But again, if you do have any questions about preparing students, I think we've already said it's a matter of practice, 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 using maths questions regularly and in a scientific context, linking maths questions in with whatever you are currently teaching. Okay, so again, MM, what I would say is in terms of being an online teacher and getting certification. Oh, Maryhan, right? Thank you, Maryhan. So if you want to become an online teacher, so what you are saying is that perhaps currently you are not a qualified teacher. Is that correct? I'm not quite sure. But obviously, if you're a qualified teacher, you don't need any separate certification to be teaching this. Right. So as far as I'm aware, I mean, obviously, I'm not sure which country you are teaching in, but I don't think you need any additional certification to teach this qualification. If you've already got, if you're already qualified as a lecturer, then I can't see any issues with you going forward and giving teaching this online. But I guess it depends on your situation and where you're actually working. Okay, let's have a look at planning the course. So Pearson provides an editable scheme of work. We'll see an excerpt from this in a moment. So you can basically customize this to suit your timetable, requirements of your school and fitting in with, how the curriculum is broken down within your school, and also the needs of the students. Some people teach this course over 1 year, some people teach it over 2 years. Term lengths are different and so on. So the scheme of work can be edited, and you can perhaps move things around, teach things in a slightly different order that suits you as a teacher, and that in a way that you think will suit your students best. So you can download this from the Pearson Qualifications website, and it's a very, very useful resource. As I said, we'll see an excerpt from this in a moment. Obviously, there's this Pearson textbook, and there is material available via a paid-for platform called Active Learn. So Active Learn contains lots of interactive resources. There are worksheets that can be edited. There are assessments that can be used, for example, for sort of tests and exams. And they all align with the content of the specification and the assessment objectives. So the best thing is to perhaps download the specification, the scheme of work, look at your sort of timetable, and then plan around that. So building opportunities for students to be sort of assessed, practical opportunities, obviously where appropriate. We've already said that there are some limitations there, opportunities for assessment and so on. So if you have the facilities and the resources to purchase Active Learn, it is a useful resource. It's one I've used when I was teaching. Lots of material, probably more material than could ever use actually, but so it gives you a good choice of resources that you can pick and choose from. So as I've already said, you can use the scheme of work to plan delivery over 1 year or 2 years, and Pearson provide a mapping document. So if you've been using a different exam board such as AQA or Cambridge International, the mapping document shows you sort of overlap between the different courses. So if you're moving from one board to another, the mapping document is quite useful. Coming back to the scheme of work, it has the specification points, and there are links to suggested lesson activities. But obviously, if you've got if you're an experienced teacher, there are activities that you often use, then you can build those into the scheme of work, should you wish to do so. And there are tasks for teachers as well as students, links to resources. So lots of useful information. Just here we go. Here we've got an example from the scheme of work. So we've got on the left-hand side the topic 9, which is internal transport. The focus is on red blood cells and blood clotting. The next column, we've got 2 points from the mark scheme, so 9.4. So 9 means it's topic 9. 0.4 is the fourth, if you like, section of topic 9, which is about red blood cells. And then 9.7 is about blood clotting. So we've got the specification points that the lesson or lessons are linked to. We've got some activities. So activities relating students matching adaptations of structures, giving an explanation, perhaps giving a presentation perhaps a PowerPoint or something on red blood cell structure and adaptations. Making a model using, in the UK, what we call plasticine and Play-Doh. Obviously, different countries use different names for modeling, clay, and so on. A diagram of blood clotting, a flow diagram. But again, if you've got activities that you have used in the past and you know work well, then you could put these in the activities column and replace some of what's already there. So the whole point is that you can use what's there, you can edit what's there, put in your own things, delete what's there, entirely up to you. We've got a reference to the human biology textbook, and then at the end two columns are additional skills that students could develop from this lesson. As we said right near the beginning, that one of the points of the specification is for students to develop skills sort of beyond the classroom, that can be used beyond the classroom, so critical thinking and problem solving and so on. So that's a typical example of a lesson from the scheme of work. So you can change sequencing, you can add in information, you can remove information, fully editable to make it useful for you as a teacher. Okay, so moving on to looking a bit more detail about mark schemes. We have looked and talked about mark schemes a little bit already. As a senior examiner, one of my jobs is to write mark schemes as we write the papers. And obviously, as I've already made hopefully quite clear, we have to work within that framework of getting the percentage of AO1, AO2, and AO3 correct as well. So we have two excerpts from a mark scheme. So the one at the top, there are 2 bullet points there. Each bullet point is worth a mark. So we have sort of the main body of the mark scheme. These are the points that the principal examiner comes up with. The next column along is what we call an additional guidance column. So here it says at the top, reject keeps body warm. So we're looking for an answer that's about trapping heat energy or preventing heat loss, and not an answer that just says it keeps the body warm. So this is a question about perhaps the skin and fat in the skin, for example. And then the one at the bottom, the example at the bottom, the question is name the independent variable, and the answer is time. So it's just a straightforward, give the name, it's time. And we're allowing units of time such as minutes or mins. So just short excerpts from a mark scheme. And here we have a slightly longer question, a short answer question worth 3 marks. It's an explanation question about time taken to digest starch. And then we've got another question, which is about infectious diseases. So the first question here, we'll be looking at a mark scheme with 3 points at least. And the one at the bottom, it's a 2 mark question, so it'd be 1 mark for diarrhea, 1 mark for malaria. Because the question says, state one way the infectious diseases, diarrhea and malaria can be prevented. So for some reason, the mark scheme for the first question hasn't come up, but for the bottom, you can see the mark scheme is split into 2 points. So the first point is all linked to diarrhea. You can see there's a huge number of points, each separated by a slash and oblique. So there's a very wide range of points that the students could write to gain the mark about diarrhea. And then again, there's quite a few points about how malaria can be prevented for the second mark, be it using insecticides or covering the arms using nets and so on. And then there's an instruction in the additional guidance column. It says ignore ORT, so that's oral rehydration therapy, because oral rehydration therapy is not prevention, it's a way of treating. So that's why you ignore it. And ignore means just imagine that they haven't written it. You just ignore it. Imagine you haven't seen it. Cover up the answer with your finger or thumb so you're not seeing it. So we just ignore it. So here's a bit more specific information about the mark scheme. So we have a column on the left, which is a question number, and then we have another column, which is the answer, which has been written in the first instance by the principal examiner. And the first iteration of the mark scheme is what the principal examiner thinks a student might respond, or how a student might respond. As I'm sure you appreciate, we don't expect students to write an answer that's worded in exactly the same way as the mark scheme, but sometimes they are and sometimes they're quite different. As long as the key points are conveyed in the answer and the answer is clear, then marks will be awarded. As I've already said, we then have an additional guidance column which gives some clarification to the examiner and obviously to you as a teacher if you're using mark schemes in your teaching. So the additional guidance column gives some clarification. So it might say accept this or allow that or so on, or as we've just seen, ignore. And then sometimes we have do not allow or reject. So sometimes if a student makes a point and then goes on to make sort of another point, which is perhaps incorrect, then we reject. So there might be something that's correct, something that's incorrect. And in that instance, we reject, we do not award a mark. So you will see the word reject quite, well, not frequently, but you'll sometimes see that word on a mark scheme. So that's where we do not give a mark for a particular point. Also, in a mark scheme, some words in the mark scheme are underlined. So if that's the case, that particular word must be given in the student's response. Sometimes we have a word in bold, which means that the word is required, or a word that means the same. In other words, a synonym is required. Sometimes we have words in smooth brackets, quite often at the beginning of a point on the mark scheme. And the reason for that is it just makes the point on the mark scheme make sense when you read it. But words in brackets are not required in the student's answer. It's more to help the examiner when they're reading the mark scheme to make sense of the whole point. And then sometimes we have words in curly brackets or phrases in curly brackets. So perhaps 2 or 3 words in a curly bracket. And what that means is that the answer must contain one of those words or one of those phrases which is within the curly brackets. So one of those words within the brackets must be present in the answer if the mark is to be awarded. So we apply sort of basic principles. If an idea is clear, then we award a mark. If vocabulary is used accurately and the point that the student makes is sort of in alignment with what the mark scheme is intended, then we would award the mark. So we sometimes will give benefit of the doubt if we to the students. But as long as the idea is you as a teacher need to make sure that students write answers clearly and concisely. They use scientific language correctly. and then that improves their chances of being awarded a mark for a particular question. So I've already said earlier on that in the International Human Biology GCSE, the questions are point-based, not level-based. So it means that even in these longer questions, there are, if it's a 6-mark question, students just have to get 6 points from the mark scheme, and they'll get full marks. So 1 point, 1 mark, etc., etc. So we don't award marks for, the quality of an answer in terms of, how well it flows and so on. The examiner is looking for specific points which are matched with the mark scheme. And if it's a 3-mark question and the student has got 3 points which are on the mark scheme, they get 3 marks. So we're not too concerned about, how does the answer flow? And I've already said we would encourage students to write short, concise answers, write answers in bullet points. It's perfectly acceptable, and it's the easiest way of being clear. We're not looking for sort of long essay style answers. As I've already said, it's about giving terminology that's precise, it's clear, and ideally well structured. Right, so I think that slide is out of context. So we've just been looking at mark schemes. I've already spoken about how some aspects of support. So we'll have a look at a little bit more about support. So as I said earlier in the presentation, the human biology website for Pearson gives you access to all sorts of information. So the specification, course materials. So there are some sample assessments, which are assessments that were produced to give examples of exams before the first assessments were actually taken. There's lots of exam materials, so past papers, mark schemes, examiners' reports. Then additional training and so on and so forth. So you can access support from the website, and there's also support available from an advisor, which I'll come back to in a moment. So from the sort of home page for human biology, you can then go on to access past papers. You may need to register. So as a teacher, you may need to register with Pearson so that you can access these materials. You can see from this image that it's showing that the documents are locked. But if you register with Pearson, then you'll be able to sign in and the documents will be unlocked. And you can see that this is an up-to-date page. It's got references to exams and mark schemes from this 2025 exam session. In terms of teaching and learning materials, you've got the course planner, exemplar material, maths and practical support, so you'd have those practical guides and so on, past training content, qualification guides, schemes of work, so you'll be able to access the editable scheme of work there, and so on. And then the slide on the right hand side is showing training content from previous events which you can also access. So lots of information available to you, lots of past papers and so on. There's also a facility called Results Plus. So if you have a group of students taking the exam, and obviously some of you might know about this, this could be new to some of you, but Results Plus gives you a detailed analysis of how well your students have performed. You can identify topics where students have done well, topics where they have done less well. You can identify whether students are doing well on, for example, AO3 questions, if they're doing well on AO2 questions. So you can pick out strengths and weaknesses, and that will then help you to perhaps think about your teaching and think about how you can change things so that students perhaps learn in a slightly different way. So it allows you to reflect on your teaching and to make improvements. You can actually use Results Plus to find out how many marks a student scored for each part of a question. You can do that for individuals, for whole classes. You can make comparisons between groups and so on. So it's a very good tool for analyzing data from exams. So the way that it works is that students sit an exam. Once the exams have been sat, the papers get scanned. So in the UK, there is a site where there is effectively a massive factory where the papers are sent and they get scanned by some complex machines so that what we then have is a digital version of the paper, and then examiners mark the papers online. Now, typically, you may not know this, but typically an examiner may not mark a complete paper. They might only mark certain parts of a question, not a whole paper. It's just the way that the online system is set up. And then when the examiners obviously mark the papers, the marks are all collated, results are issued, and then the stage 4 performance reports are shared. So you'll then, in stage 4, you can access through Results Plus, you can access data about your students' performance. But that relies on the fact that for each component of the exam, each sort of subsection of a question, we have individual marks, which means that the analysis that we can do can be quite detailed. So Results Plus is quite a powerful tool for you to use as a teacher to analyze student performance. Then another thing that Pearson provide is something called Exam Wizard. It's a really, really useful tool. Now, the slide, excuse me, the slide shows geography as an example, but that doesn't really matter. It all works in the same way. So you select the qualification that you want. You select a specification. So here that will be human biology. You select a year, so 2025, for example. Select a series, so that could be November or June for this qualification. Select a unit, so paper 1, paper 2. So what Exam Wizard allows you to do is to produce exams, tests. You can focus on a particular topic. You can focus on an assessment objective. So if you want AO1 type questions for recall, you can pick maths questions, you can pick multiple choice questions, you can focus on AO3 questions, sort of planning questions. So you can put together assessments that focus on particular styles of question. They focus on particular questions. As you go through the course, you can produce sort of assessments to check learning. So very, very useful, and it uses a massive sort of data bank of information from all the past papers. So in a relatively short space of time, you can put together your own assessments for your classes, and for different ability groups, you can produce different assessments. You can save the papers, you can edit them. So it's really, really useful. Saves a lot of time, and so you can produce, a short assessment, you can produce a longer assessment, you can choose the topics, etc. So it's very powerful, and it can even be used in conjunction with Results Plus. So something that's very, very useful from useful resource from Pearson. A question just popped up in chat. Okay, so question is, since less science students take human biology in their IGCSEs, what practicals can we do specifically to make them feel it's worth taking it? So do you mean what practicals it's worth doing in the IGCSE biology, or what practicals in general? I mean, as we've already said, within the course, students have to do the core practicals. Within IGCSE biology, again, there are core practicals. So if you mean, in your question, if you mean what practicals should we do in GCSE biology, IGCSE biology, to make them think about wanting to do human biology, then there are practicals to do with enzymes, for example, and that kind of thing that could be done. So I don't know whether you want to give me a little bit more information to supplement your question, and I'll come back to it. But thank you for asking that question. Okay, so Exam Wizard, very good resource from Pearson. One I certainly used to use a lot as a teacher. Okay, so yep. So we got obviously laboratory issues. Which one would I recommend? Absolute must-dos. So as I've said, all students doing the course have to be taught in one way or another. They have to be taught the all of the core practicals. If they can't be taught that sort of in person, then I've already said that videos and simulations are useful. Absolute must-dos. Well, obviously some of them, depending on your resources you've got available, you may not be able to do any of them. But the ones which are interesting and often work well tend to be the enzyme ones, the food test ones. They usually work well. Energy in food, because it's not usually the equipment needed for energy in food isn't too sort of demanding. It's just a matter of having a container, a thermometer to heat some water, and something to burn some food with. So they're quite straightforward. But the short answer to your question is all of them are absolute must-dos because they're part of the specification. And if you're not able to teach any of them directly firsthand, then use a video, use a demonstration. So like I said, I've got to say that they're all must-dos, but in terms of it's a matter of what you can actually, the resources you can get hold of in Bangladesh. Some are obviously easy to do without a specific laboratory. You don't necessarily need a laboratory to find energy in food. You don't need a laboratory to compare how much carbon dioxide is in sort of inhaled and exhaled air. You don't need a laboratory to measure pulse rate. You don't need a laboratory to measure breathing rate and so on. So there are quite a few where you don't need a laboratory at all. Hopefully I've given you a satisfactory answer. If I haven't, please put something else into the chat. But a good question, and thank you. Okay, another facility from Pearson is access to scripts. So when a student is entered for the exam, obviously a fee is paid, and part of that fee means that once the results have been issued, students can you can access scripts. So you can actually look at the marked script online, or you could choose to download a PDF of that script. So again, you can, whereas Results Plus allows you to analyze data, look at the marks, access to scripts means that you can actually look at a marked script. So again, you can look at how well a student has answered a particular question. You can see how well a student or a group of students have done on particular types of question. So you can see the language used by your students, which will then perhaps think, okay, these students aren't explaining something very well. I need to make sure they use better language. So you can look at the mark script, you can look at the mark scheme, because the mark schemes are available to you as well from when the results are published. So this is part of the, as a result of paying an entry fee, you get access to scripts. So again, you can sort of find out more about that from the Pearson website, as you can see from the additional information in this slide. So there's clear information about how to access it, what you can use it for. And Pearson is the only awarding body that provides access to this as part of your qualification fee. So other exam boards might charge a separate fee for accessing scripts, whereas with Pearson, you pay the entry fee, the qualification entry fee for the students, and you then get access to the scripts when the results have been published. So very useful, and it's particularly useful if you find perhaps a student hasn't done very well, and you want to think, okay, I might want to have the mark reviewed. If you yourself look as a teacher at the mark script, you might think, well, actually, this looks to have been marked correctly. I can't see any issues. There is no point in paying an additional fee to have the mark reviewed. So it can, in the long run, save money. Obviously, other resources, if you're teaching the course, then you might obviously find it useful for the students to have this human biology student book. And there are sort of updated versions of this. But it's good. It contains obviously all the information that the students require, ties in with the specification, and you can find more about it, and you can get examples or samples from the book. If you see there's a link at the bottom of this slide. So we have in Pearson, we have subject partners. Now, one of the previous slide, there was another sort of subject partner, an expert, who was shown on one of the slides. But here we have Jonathan Wong, International Sciences. You'll see there's a generic email, teachingscience@pearson.com. So you can sign up through this to receive updates, regular email updates, certainly monthly about the subject, receive support. You can email the Teaching Science email address. if you have specific questions about the course. So that's their job. They're there to answer your questions. So do not hesitate to get in touch via email if you have any questions, whether it's about starting the course or whatever it might be. It's their job to provide you with the answers. So we've got, as you can see, there's a wide range of resources available to you as a teacher, sort of online, and the sort of the personal contact using using email. And you can see there's even a telephone number there for you as well, should you want to contact them directly. Just appreciating that I think the times given would be UK time rather than anything else. Okay, so got another question in chat. So about the 9-1 grading. Okay, I'll answer the second part of the question first. So a PGCSE certificate important to becoming a Pearson qualified teacher. Well, certainly in the UK, teachers have a PGCE or similar qualification. I mean, as an international teacher, I'm not 100% sure what kind of additional qualifications you might need, but that is exactly the kind of question that you could ask the subject partner, so Jonathan Wong in this example. It's exactly the kind of question you could ask him, because I can't give you a specific answer there. In terms of the 9-1 grading system, so basically, as we know from earlier on in the presentation, the total number of marks is 180. So that's for the two papers combined, paper 1 and paper 2 combined. So in terms of the way the grading works, so for example, a certain number of marks would be needed for, say, to achieve grade 5. A certain number of marks is needed to achieve grade 9. So clearly, so we have grade boundaries. So if you get X number of marks, you get grade 1. And then if you get sort of Y marks, you get grade 2, and so on, and so on, and so on. So there are grade boundaries for each of the grades. And if you get sufficient marks to cross a boundary, you get that grade. So clearly for grade 9, the grade boundary out of 180 it would be very high. I can't give you an exact figure. The figures vary sort of exam year on exam year. They vary a little bit, but that's basically the way it works. So the more marks you get, the higher the grade you get. Okay, so the marks and the grading, so the raw marks, in other words, the marks that the students get on each paper, they're added together, and then the total mark is used to arrive at the final grade. So the more marks a student gets, the better. So they might do much better on one paper than another, but the total they get is the mark that is the mark that is used to arrive at the final grade. Hopefully that helps. Another good question. Right, do we have any other questions? So I hope that the presentation has been useful to you. There's clearly a very, very large amount of information that I've been trying to get across. As you know, there'll be there's a digital, well, a copy of the presentation available to you, and also there should be a recording of the presentation available to you as well. I'll just give a moment for you to ask any questions should you have one. We've sort of just nicely got towards the end of our allotted time. I hope it's been useful to you. So, like I said, if you do have any questions or comments, please put them into chat. So, okay, so I think in terms of if you like, the grade boundaries, the marks that you need for each grade, I think I can't give you specific directions of where to go, but I think you can find some of that information from the Pearson website, but also that will be another question perhaps to ask the subject advisor using that email that was on one of the previous slides that we've just been looking at. And sort of A star, that's sort of equivalent to grade 8, grade 9. Sometimes we used to say that grade 9 was like a super A star. So, okay. And thanks for the comment that's just come in. That's much appreciated. And hopefully, as somebody commented a little while ago, clearly the number of students doing human biology IGCSE is smaller than the IGCSE biology, but clearly it's another option available to you in schools. It could be used perhaps as a standalone qualification for science. Obviously, it's used in different ways in different schools, different countries. Any other comments or questions? If there aren't, I would like to say thank you. I will leave the chat open for a little bit longer. Otherwise, I wish you well with the rest of your day. I do appreciate it. Could be late in the day for some of you. Say in the UK, it's just approaching midday. But clearly, where some of you are listening from and joining from could be much, much later in the day. So thank you very much for your participation. You should receive an e-certificate at some point in the near future to confirm your participation in this event. In terms of sharing LinkedIn profiles to stay connected, I don't know is the short answer to that question. I don't know whether our Pearson host can answer that question. I'm not sure. I'll just give opportunity for our Pearson host to perhaps think about that. Thank you for the comments that are coming through. It's clear that obviously some of you are using sort of online teaching as a basis of your work. Clearly, everybody involved in international GCSEs is their experiences are different. I don't have a LinkedIn email contact, but you could always, if you have any additional questions, you could access me via by asking the International Pearson team. Okay, unless we have any additional comments, I'd like to just say again, thank you very much for participating. And I will close the training. So like I said, I wish you well for the rest of your day, whatever time of day that might be and whatever work you may have left to do. So goodbye, and hopefully we may, if you like, meet again on a future online training session. Yeah, so there will be certification. You should receive an e-certificate at some point in the near future. Again, I think our Pearson hosts will perhaps confirm that. If you don't receive certification, then you can again contact the Pearson representative, but you should automatically be sent, if you registered prior to the event, you should automatically be sent an e-certificate.
I just want to confirm that is correct. That they will be receiving the e-certification after the training. So it should be in the email that they used to register for the training.
Perfect. Thank you, Jaira. Thank you. Okay, everyone. So goodbye and thank you.